Hello everyone. We will talk about sex determination in honey bee. I have already discussed about the sex determination in human beings, birds and insect in the last lesson. But one more topic is there, that is how the sex is determined in honey bees. Actually, in honeybees, the sex is determined also by the number of the chromosomes, that how many chromosomes are present in the individual body, and accordingly the sex is determined. The sex determination in honeybees is also known as haplodiploid sex determination. Let us see that how the sex is being determined. In this case, you can see here the female honeybee, that is queen or you can say worker, has total 32 numbers of the chromosomes. If there is a question that how many chromosomes are found in the female honeybee, 32 chromosomes, and in males, that is called drones. In the individual male or in the drone, the total number of chromosomes are 16. And the drones, they are haploid. Here the males are always haploid, having how many number of the chromosomes? 16, while the female, that is the worker, the female is also known as the worker or the queen, they have total 32 number of the chromosomes. The female, or you can say the queen, either worker, is produced, the female is produced by the union of, by the fertilization of the egg, by the union of the male and female gametes. When the male and female gametes, that is the egg and sperm, both are fused together, the egg has how many number of the chromosomes? 16, while the sperm also has how many number of the chromosomes? 16. So the egg and sperm, when both they fuse together, the fertilized egg always develops into the female. It is very, very important. When the process of fertilization, keep in your mind, when the process of fertilization takes place in the case of honeybees, when the sperm and egg, both they are fused together, it forms the zygote, and zygote always develops into the female. And if the egg is unfertilized, suppose the egg is not going to fuse with the sperm, the unfertilized egg is produced into the male drones. That's why the male has how many number of the chromosomes? 16. You can see here the female, it means how many number of the chromosomes are there? 32. And during the process of gamete formation, because the 32, it is the diploid number of the chromosome. It is diploid number of the chromosome. So during the process of gamete formation, during the process of the gamete is produced by the process of meiosis division. During the process of the meiosis, the gametes are formed. You can see here the egg eggs are produced, and each egg has how many number of the chromosomes? 16. And if the egg, suppose, is not going to fertilize with the sperm, then it will produce the male offspring. And if the egg is going to fuse with the sperm, which is produced by the male during the process of mitosis, I have written here, male has how many number of the chromosomes? 16. It is always haploid. So male produces the male gamete by the process of mitosis only. And that's why the sperm has how many number of the chromosome? 16. This one is the sperm. And when the sperm and egg, both they fuse together, they form what? The zygote, which develops into the female. The unfertilized egg, if the egg is not going to fertilize, the unfertilized egg, it means it develops into the male drones. And if the egg develops into the male directly without fertilization, this process is called parthenogenesis. So you can say, if the egg is unfertilized, it can produce parthenogenetically the male, and male has 16 number of the chromosome. Male produces the sperms, and each sperm has 16 number of the chromosome. The sperms are produced by the process of mitosis. When the sperm is fused with the egg, again the zygote is formed, which develops into the female. So it is very, very important. Once again, you have to keep in your mind that if the egg is fertilized with the sperm, then the female offspring will be produced, and if egg is unfertilized, then it will produce a male offspring, or you can say male drone. I have written here some of the points. Offspring formed from the union of sperm and egg. Union means when the fertilization takes place. Offspring formed from the union of the sperm and egg develops as female. It may be the queen, either worker. Second, unfertilized egg. If the egg is not going to fertilize, the unfertilized egg develops as a male, and that is called drone. And this unfertilized egg, if develops into the male drones, this process is called parthenogenesis. We will discuss later on about parthenogenesis in detail. That parthenogenesis is the process. It not takes place only in the case of the honeybee, but overall the process of parthenogenesis, it takes place in the plants also, in honeybees also, in some of the rotifers also, and the in lizard also, in turkey birds also, the process of. So parthenogenesis is the process in which egg directly develops into the young ones without fertilization. And here the egg develops into the male drone without fertilization, and that's why the male drone has how many number of the chromosomes? 16. And male drone, it's very, very important, produces the sperms by the process of mitosis. And the sperm, when fuses with the egg, it forms the zygote having 32 number of chromosomes, and this fertilized egg, that is zygote, develops into the female offspring. So the here I have written, male produces sperms by the process of mitosis. They don't have father. It's very, very important. In the case of honeybees, the male, that is drone, don't have the father. Why the male don't have the father? Because male is produced by unfertilized egg, and egg is always contributed by mother, and that's why the male don't have the father. I have written don't have the father, and thus cannot have son. It's also very important. Why the male don't have the son? Because when the male produces the sperm, when the sperm fuses with the egg, it forms the zygote, and as you know, zygote always develops into the female offspring, and that's why the male don't have the son even. So it's very, very important. Male honeybee don't have the father and don't have the son, but they have the grandfather and can have the grandson. Definitely the grandfather and grandson will have in the case of the honeybee. So overall, in the case of honeybee, the process of sex determination is very special, is very unique, and it is very, very important regarding your examination, regarding the competitive examination also. Last year, there was a question that how many number of the chromosomes are present in the male honeybee in the drone, that is 16, and how many chromosomes are present in the female honeybee, 32. And you can see your book, you can make this diagram also, and you can revise the same. Now, the next topic in this chapter is mutation. I have written here about the mutation. What do you mean by mutation? Anyone? What do you mean by mutation? The mutation is the process of the change of the genetic material. Mutation is the sudden change in the structure of the DNA, in the structure of the gene. As you know, our gene, so many genes, they are present on our chromosomes. The chromosomes are the bearer of the genes. And whatsoever the genotypes are to be there, genotypes means the genetic setup. The genetic setup, the genetic structure, the genes, they are responsible for our phenotypic expression. Means whatsoever the phenotype is to be there, means our outlook, that is our skin color, our hairstyle, the color of our eye. Hairstyle means some of the hairs they are straight, some of the hairs they are curly. The color of the eye, the human skin, your height. Not only your phenotypic expression, even inside the body also. Most of the biological activities takes place under the supervision of the genes. Means the genotype controls our phenotype. How you can say that our genotypes, they are responsible for our phenotypic expression. Suppose your height, you are tall, means the gene is to be there, and that gene controls your height, your tallness. Suppose you are dwarf, maybe the gene, as per Mendel, you have already studied that the tallness, the dwarfness, the shape of the seed, the color of the seed, the position of the flowers, means all the traits, all the characteristics are controlled by the genes. At the time of Mendel, he used the term factors. The factors are responsible for the phenotypic expression. The same like in our body also, means whatsoever our phenotype, our look is to be there, the gene is responsible for that particular character. And if any change is to be there, suppose if the gene is going to be mutated, if the gene is going to be changed suddenly, our phenotype will be affected. As you know, the genes, first of all, inside our body, they produce some of the proteins. Our body is made up of mainly proteins. So the genes at the molecular level, I will teach you in the next chapter. The genes, they control the synthesis of, they produce different types of the proteins, and these proteins ultimately they construct our body cells and tissues, and accordingly we are able to express our phenotype. And if the gene is going to be changed, that particular protein will not be synthesized, maybe the enzymes are not going to be synthesized which are responsible for the process of metabolism, and overall our phenotypic expression would be affected. If your body shape is designed in a particular way, means the genes they are working. If the genes they are going to be mutated, they are going to be changed, definitely your look will be also affected. For example, we have 5 fingers, but in some of the cases, like in the case of problem in pentadactyly, some people they are having how many fingers? 6 fingers. Maybe the gene which controls or which are responsible for these 5 fingers may be mutated, may be changed, and because of the change in that particular gene, in place of the 5, there are 6 fingers. I will teach you so many abnormalities like hemophilia, color blindness, alkaptonuria, phenylketonuria, thalassemia. So many disorders are there, and we will discuss in detail in this chapter also. Some of the diseases like Down syndrome, Klinefelter syndrome, we have to study. But in this chapter, in your NCERT book, the mutation has been given in there is a brief introduction of the mutation, because at this level you will not understand the mutation in detail till you are going to cover the next chapter, that is molecular genetics. So here in this chapter, in mutation, I will give you a very brief introduction, so you will able to know about some of the Mendelian as well as chromosomal disorders. So overall, mutation, what is mutation? Mutation is the process of the change in the structure of DNA. The mutation is of 2 types, gene mutation and chromosomal mutation. The gene mutation is a very small mutation. It takes place at the gene level, not in the whole structure of the chromosomes. As you know, suppose these are the chromosomes, and the chromosomes are the bearer of genes. So many genes they are located on the chromosomes. Even a single chromosome may have nearly 100 thousands of the genes, they may be present. In the gene mutation, we are not talking about that how the chromosomes they are going to be changed. If the mutation takes place in a particular gene which is located on the chromosome, this is called what? Gene mutation. So the mutation which takes place in the gene, this is called gene mutation. And gene mutation is further divided into the 2 parts, that is point mutation and Frameshift mutation. Point mutation, I will teach you in the next chapter. Even a particular gene also is made up of so many units, the nucleotides. I will teach you in detail in the second chapter. Adenine, guanine, cytosine, thymine. In the case of RNA, it is uracil. At the very molecular level, if any base, that is adenine, either guanine or cytosine or thymine, it is going to be replaced by some other base, it is said to be what? Point mutation. In point mutation, purine, one purine may be changed into the another purine, or the purine may be changed into the pyrimidine. The point mutation is also transition, transversion. You will be able to know in detail in the next chapter. So if the base, suppose in a particular gene, there is the base and that is called GUG, and in the place of, suppose there is a base GUG and this U I am going to replace here by C. A single base is going to be, in both of the places G are there, but a single base may be changed and it can affect the process of metabolism, the protein synthesis, and finally it may be the cause of the disorder. And there is a very good example of the point mutation, that is sickle-cell anemia. I will teach you in the next topic. In the Mendelian disorder, we will discuss about the sickle-cell anemia. The sickle-cell anemia is a very good example of point mutation. I will tell you in detail that how a change in the single base is the cause of the point mutation. Here I think no need to discuss about the sickle-cell anemia. It is included in the Mendelian disorder. So overall, point mutation is a type of gene mutation in which a single base is changed into the another base, and the example of the point mutation is the sickle-cell anemia. I request you all to make these small notes. You just see, no need to go beyond the NCERT book, just you take the NCERT book and you find out the definition, and the same definition you note down in your fair copy. That will be sufficient for your 12th examination. It is already given that is what is point mutation. Give one example of the point mutation, that is sickle-cell anemia. I will teach, definitely we will discuss in detail about sickle-cell anemia, that what is sickle-cell anemia, how a single base, what is the base, how a change in the base is the cause of this disease. Don't worry, I will discuss step by step into the next topic. But here in this mutation topic, the definition is very, very important, that what is mutation, what are the types of the mutation, what is gene mutation, what is point mutation. These definitions are very, very important. The second, Frameshift mutation. The Frameshift mutation, in Frameshift mutation, it is further divided into the two types. There may be the insertion or deletion of the bases. Insertion or deletion. Suppose a particular gene is there, and in this gene, these are the sequence. And suppose one base is going to be inserted in between here, the C and T, and this is G, then the whole structure will be changed. You don't insert here. I am going to insert here the G. Now A, G, C, G, C, T, A, C, G, G, C, G, like this. Earlier the codon was ACG, now it is AGC. The second codon is CTA, here GCT. Actually, in this chapter you are not able to understand about the codon because all these things are included into the next chapter. But overall, if in a gene so many bases they are arranged, and suppose a particular base is going to be inserted, due to the insertion of a particular base, the whole frame, it is going to be changed. I will teach you in the next topic about the codon, that a particular codon codes for a particular amino acid for the protein synthesis. And all these codons, they are well specified, that ACG or CTA or CG, CGC, it will code a particular amino acid. But due to the insertion of the base, the whole frame is going to be changed. And some unwanted amino acids, they are going to be coded, which further participate in the protein synthesis, and overall it can affect the phenotypic expression of an individual. In the deletion also, if the base is going to be deleted, suppose the T is going to be deleted, it will also affect the whole frame, the whole structure of the gene. So in the Frameshift mutation, due to the insertion or due to the deletion of one or two bases, the whole frame, the whole structure of the gene is going to be changed, and that can cause different problems or disorders into the human beings in an organism. Now let us talk about chromosomal disorder. What do you mean by the chromosomal disorder? The chromosomal disorder is the abnormalities, it is also known as chromosomal aberration, in which the number of the chromosomes, either one or two chromosomes, they are going to be added into the normal set of the chromosome. Either one or two chromosomes, they are going to be missed from the basic set of the chromosomes. For example, how many chromosomes they are present in our body? We are diploid organism, and the total number of the chromosomes are 46. 2n equal to 46. Means we have two sets of the chromosomes like this. 1, 2, 3, 4, like this. 23 here and 23 here. And if one or two chromosomes, they are going to be added, it will be the chromosomal disorder. Actually here the chromosomal disorder, or you can say the chromosomal mutation, is divided into the two types, aneuploidy, and the second one is the polyploidy. It's very, very important. What do you mean by aneuploidy? Aneuploidy is the case, is the condition in which one or two chromosomes, they are added or deleted from the basic sets of the chromosomes. For example, our basic sets of the chromosomes, we are diploid 46, 2n equal to 46, and suppose one chromosome is going to be added, means if the condition is going to be 47, then it will be the cause of a syndrome, and that is called Klinefelter Syndrome. In Klinefelter Syndrome, 2n plus 1. The aneuploidy is further divided into the two types, monosomic and trisomic. In this is the formula of trisomic, 2n plus 1, and in monosomic, the formula is 2n minus 1. Our basic set number of the 2n equal to, this is the basic set of the chromosome, 2n, basic set, and due to the mutation, due to the chromosomal mutation, one chromosome is going to be added here, and this condition is called trisomic. Means how many total number of the chromosomes will be there in trisomic condition? 47. As you know, 2n, the normal number of the chromosomes are 46. These are the basic chromosomes in our cell, 46. And if one more is going to be added due to any cause, then the total number will be 46. And because of the increase of additional number of the chromosome, it may be the cause of the problem, disorders. And we will discuss about two syndromes, that is Klinefelter Syndrome. And in Klinefelter Syndrome, we will discuss in this chapter later on in chromosomal disorder, the total number of the chromosomes, it becomes 47. There is the extra X chromosome. The Klinefelter Syndrome, 44 plus XXY. Normally in male, how many chromosomes? 44 autosomes. And these two are sex. So this is the normal condition. But due to the addition of one extra X chromosome, the disease is caused, and that is called Klinefelter syndrome. In Down syndrome also, the total number in Down syndrome, I will teach you later on, the total number of chromosomes, it becomes what? 47. But the trisomy is caused on 21st number of the chromosome. The 21st one, it becomes trisomic, 2n plus 1. So many syndromes are there. Regarding the trisomic, Klinefelter syndrome, Down syndrome, Edwards syndrome. If 18th it becomes trisomic, it causes Edwards syndrome. If 13th, 14th, 15th, they become trisomic, it may be the cause of Patau syndromes. Means the syndromes, there are different types. But overall, in aneuploidy, what is going on? In aneuploidy is divided into the two types, monosomic and trisomic. In trisomic, one chromosome is going to be added into the basic sets of the chromosome. And here I have given the example 2n plus 1. This is the formula of the trisomic condition. And the diseases which are related to the Klinefelter syndrome, which are included in your NCERT book, the two diseases are there, Klinefelter later on. Now monosomic, what is the formula of monosomic? 2n minus 1. The basic set number of the chromosomes are 46. 2n equal to 46. And suppose one number of the chromosome is going to be removed, then the total number of the chromosomes, it will be 45. And the disease which is associated with the monosomic condition is Turner syndrome. I will teach you later on about the Turner syndrome. The formula of Turner syndrome is like this: 44 plus X0. As you know, in the female, the normal karyotype is 44 plus XX, but 1X, if it is going to be missed, then the total number of the chromosome, it will be 45. And if any one or any individual has 45 number of the chromosomes, then what will happen? That will be that will be affected from the syndrome, Turner syndrome. And if anybody has 47 number of the chromosomes, what kind of disease they will have? If X chromosome, it is going to be added additionally, that will be the Klinefelter syndrome. And if 21st it becomes trisomic, that will be the Down syndrome. All the syndromes, the diseases, we will discuss later on in this chapter. in the next topic I will teach you. But overall, in the aneuploidy, not the whole set of the chromosomes they are going to be increased, either decreased, but one chromosome or maybe the two chromosomes. In your textbook, one chromosome, 2n plus 2n minus 1, these two examples are there. But one or two chromosomes may be added into the basic sets of the chromosome, or may be deleted, may be absent, like in the case of monosomic. In monosomic, one or two becomes less, and in trisomic, it becomes more in the basic set of the chromosomes. How the process of aneuploidy it takes place? The homologous chromosomes fail to segregate during the process of meiosis. As you know, why? What is the cause of aneuploidy? How it happens that some chromosomes they are going to be added or may be removed, deleted. Why there is the disturbance? Why the process of aneuploidy is caused? And aneuploidy is caused, normally what happens? You have already studied in class 11 that during the process of meiosis division in anaphase condition, the paired chromosomes, they are separated to each other. In a particular stage in anaphase, in the metaphase, all the paired chromosomes, they lie in the center in the equatorial plane, and the paired chromosomes, they separate to each other. So 23 it comes here, 23 it comes here. But suppose if there is any error takes place during the segregation, if the paired chromosomes they fail to segregate to each other properly in a pair, it can cause aneuploidy. And when the chromosome, there is a particular term not given in your NCERT book, that is called non-disjunction. What is non-disjunction? Non-disjunction is the condition, is the phase in which the paired chromosomes fail to segregate properly during the gamete formation. And when the non-disjunction takes place, or you can say when the chromosomes, the paired chromosomes, they fail to segregate properly, this condition, the aneuploidy condition, is created. It comes. Now the second one is the polyploidy. What do you mean by polyploidy? In the case of polyploidy, the whole sets of the chromosomes, they are going to be increased in the normal, in the basic set of the chromosome. Suppose our basic sets of the chromosome, we are diploid organism, and suppose one more set is going to be increased here. It becomes what? Earlier it was diploid, and if the three sets are to be there, this is the diploid condition 2n. If the three sets are there, one set is going to be added in the normal set, it becomes 3n. If four sets are there, this is triploid condition, tetraploid. If five sets are there, it is called polyploid. More than two sets generally, it is called polyploidy condition. Tetraploid, pentaploid, hexaploid, the wheat which we consume, do you know? It's a hexaploid plant. It has how many? Total number of the chromosomes are 42, and in each set 7 chromosomes are there. So 6n, the ploidy of the wheat plant is hexaploid. Actually, the case of this polyploidy, this aneuploidy occurs in the human beings, but the polyploidy condition has been not reported in the case of human beings. It's very major mutation, And this polyploidy condition has been created in the plants naturally. Sometimes it is induced artificially to get the desired product, but the polyploidy conditions are found mainly in the plants. And this polyploidy condition in the plants, they may be useful, and that polyploidy conditions may be the cause of the more yield, may be beneficial in the case of the plants. But what differences are there in aneuploidy once again and polyploidy? In aneuploidy, the whole set is not going to be changed. Just 1 or 2 chromosomes, they are going to be added or may be deleted into the basic sets of the chromosomes. But in the case of polyploidy, the whole set, they are going to be changed. If the normal set is diploid, it may be triploid, tetraploid, pentaploid, hexaploid, and more than 2 sets of the chromosomes. This condition is called polyploidy, and the polyploidy is reported in the case of the plants. But aneuploidy, it occurs in the human beings sometimes due to the error during the process of cell division, and it is harmful for us, for animals, for human beings. But polyploidy may be useful. Sometimes in the plants, a particular chemical is to be there that is called colchicine, and that colchicine is used to increase the polyploidy level to get the desired product. So that's all about aneuploidy and polyploidy. Overall, mutation in this chapter, the simple definitions are very important. That what is mutation? The mutation is the process of the change in the structure of the DNA. And the chemicals, or you can say the physical agents, or you can say the substances which are the cause of the mutation, they are called mutagens. It's also very important. I couldn't explain earlier. So mutation is the process of the change in the structure of the DNA, and mutagens are the substances which are the cause of the mutation, and the mutagens may be the physical agents or the chemical agents. Some of the rays are there, ultraviolet rays, gamma rays, so many rays, they act as the mutagens. They may be the cause of the mutation. And some of the chemicals, definitely you might be aware, the chemicals, they can also change in the structure of the DNA, and these are called chemical mutagens. So mutation is the change in the structure of DNA, and mutagens are the substances which are the cause of mutation. So that's all regarding this mutation, and today I also covered the sex determination in honeybees. Please note down some of the questions related to the topics which I have been taught today. Question number 1. How many number of chromosomes are found? How many numbers of chromosomes are found in male drone? In male drone. Question number 2. How the sex is determined? How the sex is determined in honeybees? Explain. Explain. Question number 3. What is mutation? What is mutation? What is mutation? Write its types also. Next, differentiate between gene mutation and chromosomal mutation. Differentiate between gene mutation and chromosomal mutation. And the last question is explain aneuploidy and polyploidy. Explain aneuploidy and polyploidy with the help of examples. Explain aneuploidy and polyploidy with the help of examples. This I couldn't explain. What is the cause of the polyploidy? Here I have written, as I taught you, the cause of the aneuploidy is the non-disjunction in which the homologous chromosomes fail to segregate properly during the process of meiosis. And how the process of polyploidy takes place? The failure of the cytokinesis after telophase stage. This is the condition. During the process of, as you know, telophase, just after the telophase, the process of cytokinesis takes place. In cytokinesis, the cell is going to be divided into the two parts, but when the proper cytokinesis doesn't take place after the telophase formation, this condition, the polyploidy, is created. The causes are very, very important. These 2 points you will underline in your NCERT book. You will write that what is the cause of aneuploidy and what is the cause of polyploidy. So that's all for today. Thanks to each and everyone.